for the very kind introduction. I'd like to thank the organizers of this meeting to invite me to this wonderful opportunity for exchanges. So I'm here to give a talk on common disease genetics from the perspective of Hong Kong. So this is a view of Hong Kong Island, Victoria Harbour. So Hong Kong is a very vibrant city with a population of 7.5 million people. And we have 8 universities. And my university is the University of Hong Kong, which is on the west side of the Hong Kong Island. And our government spending on research is about 0.73 of our GDP, which is still, we feel, a bit low. And we've been trying to get the government to increase the spending on research. So in terms of the uptake of genomics in Hong Kong, I think we are a little bit slow in picking up this, and the patient organizations, patients with rare diseases, have been trying to get the government and the hospital authority to make molecular diagnosis more accessible to them. And finally, our government is now going to invest in this area. So our chief executive, in her policy address last year, announced that the Hong Kong Genome Project will be started. This is on the recommendation of a steering committee, which I'm a part of. So the objectives of the Hong Kong Genome Project is to enhance clinical application of genomic medicine to benefit patients, and also to promote research in genomic medicine so that it can help the development of medical services in Hong Kong. And it will be in 2 phases. In the pilot phase, 2,000 whole genomes will be sequenced, and these will be mostly on undiagnosed genetic disorders and cancers. And then in the main phase, an additional 18,000 cases will be sequenced. So again, focusing on these disorders, but also other disorders. And in terms of implementation, our government have looked at the different genome projects across the world, and they visited the UK and talked to people at Genomic England. And so it's very much modeled on the Genomic England model. So there will be, they will form a company which is entirely government owned, and putative name will be Hong Kong Genomic Institute. But of course, this company will have to work very closely with partners. So key partners will include the Bureau for Food and Health, Department of Health, Hospital Authority, which looks after all the public hospitals in Hong Kong, the universities, the private hospitals, and other research and development institutions and companies. So in the University of Hong Kong, we established a Center for Genomic Sciences shortly after the completion of the Human Genome Project. So at that time, actually, the university tried to recruit Professor Lap Chi Chui to be the director, but we subsequently recruited him to be our vice chancellor. But nevertheless, the center was established, and now we have a team of about 40 scientists, technicians, IT, bioinformatics people, and so on, to provide this core support. And we are now based in this building, the Jockey Club building for interdisciplinary research, and we have a fairly comprehensive range of technologies, including technologies for high-throughput sequencing. So Pacific Bio, we established about 2 years ago, and last year we added the NovaSeq. And also to deal with the large amounts of data generated, we have established a high-performance computer cluster. So this is our website, and these are the main areas of core services that we provide. And about 80% of our jobs are internal, so serving PIs from the University of Hong Kong, and the other 20% other institutions, mostly in Hong Kong, but also overseas as well. So I'm now going to switch to give you a sort of overview of the genetics of common diseases, but concentrating mostly on work in my group and in Hong Kong. So I think most of this conference have been on rare disorders. So indeed, rare genetic disorders are probably where genomics will have the greatest impact in the short term. So I think in the longer term, genomics is also going to have an impact on more complex, common diseases. So unlike single gene disorders, which are very distinctive, common diseases are usually more variable, and they often reflect an underlying pathology which is continuous rather than discrete. So even though myocardial infarction is a discrete event, but underlying it is the narrowing of the coronary arteries, which is continuous in nature. So there are different severities of narrowing, and this underlying continuous pathology is usually determined by multiple genetic and environmental factors. And this is why the multifactorial model is most appropriate for common diseases. And this is an analogy by a group from Toronto. So if you consider the genetic and environmental factors as these marbles, then an individual will have a certain amount of these factors, and when these total burden of factors exceed or reaches a certain threshold level, then the disease develops. So this is a so-called reliability threshold model. Reliability is the total amount of genetic and environmental factors that the individual has, and the threshold value is the level of liability at which the disorder occurs. And in classical genetics, we can use twin studies to estimate the relative contribution of genetic and environmental factors. So the proportion, contribution from genetic factors is called the heritability. And twin studies on different common diseases have estimated the heritability of these disorders, and it ranges from very low heritability, such as for uterine cancer, cervical cancer, to very high levels of heritability. Yeah, so to diabetes and so on. Of course, genetics has now moved on to the molecular era. So instead of just estimating the total genetic contribution, we can now actually look for the specific genes and the specific genetic factors that predispose to diseases. And modern technologies now allow us to do this in a very comprehensive and powerful way. And for common variants, the variants with a frequency of 5% or more, these can be now pretty much comprehensively measured using SNP arrays, such as the Illumina iScan platform here. So this is a result of international projects such as the HapMap and 1000 Genome Project, which allow us now to have arrays that cover over 90% of all common variants. For rare variants, variants with frequency of less than 1%, many of them remain unknown, and so it is necessary to use high throughput sequencing technology. And so the NovaSeq is the platform with the highest throughput, but despite the reduction in cost, this is still much more expensive than the SNP arrays. So for common disorders, in the last 10 years or so, most of the efforts have been genome-wide association studies using the SNP arrays, because SNP array being so much cheaper, we can study much larger samples. So the principle of genome-wide association studies is very simple. We simply collect large samples of patients and controls and use the SNP arrays to measure the allele frequencies and statistically try to identify SNPs where the frequencies are significantly different between cases and controls. So these association signals are then represented in Manhattan plots and so on. And so this can be done using software such as Plink or other programs. So a couple of points is that because of the very large number of SNPs that are examined, so often many millions, the statistical threshold, the p-value needed to reach significance is very small, so typically 5 times 10 to the minus 8. And therefore, partly because of this, we need very large sample size in order to reach statistical power. And this is why, in recent years, research has moved on to international consortia. So no single group can collect large enough sample size for this to be powerful. So researchers from different centers and different countries have decided to join forces and perform meta-analysis of the data combined from all these centers. And this is an example on schizophrenia. So as a psychiatrist, I've been involved in this consortium, this Psychiatric Genomics Consortium. So this is a landmark paper published in 2014, and you can see that in total it contains almost 40,000 cases, and I think almost 45,000 controls, so very large samples. And with this, we identified 108 independent genetic loci associated with schizophrenia. And this is really quite typical in the field of common disease genetics. So there are many other diseases, Crohn's disease, diabetes, and so on, which have really experienced the same thing. And as the sample size increased, many loci were identified. So by 2017, 2 years ago, Peter Vischer reviewed all the genome-wide associations studied at that time, and there were 10,000 significant associations identified for common diseases. And going slightly more recently, more forward, so by the first quarter 2018, there were 78,000 robust associations for 300 complex diseases. I think if I were to do this now, probably even more. But what's the use of this, of all these genetic associations? Well, they may represent drug targets. And the evidence for this is that some of our non-drug targets coincide with these GWAS hits. So for schizophrenia, the most, or pretty much all drugs that we currently use to treat schizophrenia act by blocking dopamine D2 receptors. And indeed, one of the 108 GWAS hits was on the dopamine D2 receptor gene. Another example is for the treatment of lipids. So we all know that statins are used to treat hyperlipidemia, and statins typically reduces LDL by about 14 to 17 units. And the target of statin, which is this gene, HNGCR, is one of the GWAS hits for lipids. Of course, the effect size is small compared to the effects of statin. And this is because the common variant has a limited impact on gene expression. It doesn't mean that drugs will have the same effect size. So what this means is that even though the effect size of a GWAS is small, it doesn't mean that if you were to interfere with that gene, the effect size will be equally small. And together with a team from GSK, the company, we looked at to see whether the GWAS results have a more sort of general relationship to the success of drug development. And so they look at drugs in various stages of development, from the earliest preclinical stage to the final sort of clinical stage. And they found that the drugs in the final stages are more likely to have genetic support than the drugs at the earlier stages. So the interpretation of that is that the drugs with genetic evidence are more likely to pass through these different stages successfully. Another important use of the genetic findings is to try to obtain insight into the disease mechanisms and the biology of disease. So for schizophrenia, analyzing the function of the 108 GWAS hits reveal that many of these hits coincide with genes that serve one of these three areas of function: the synaptic function, immune function, and historegulation. And of course, synaptic function perhaps is not surprising, schizophrenia being a psychiatric disorder. But these are the two functions, histone and immune, perhaps are somewhat less expected. And the importance of epigenetics is also supported by a study that we published in 2014. So we found association at chromosome XQ28, and this region contains this MECP2 gene, which is important in regulating DNA methylation. So, and there is also converging evidence between GWAS studies, which concentrate on common variants, and studies on copy number variants, which tend to be less common than the single nucleotide polymorphism. And an example is just what I just mentioned, our GWAS finding in HQ28. And in this very comprehensive study of copy number variants on 41,000 subjects, XQ28 also comes up as being enriched for CNVs associated with schizophrenia. And there are other genes that are implicated by the CNV analysis from this study. So for example, these molecules that are involved in the presynaptic terminal of the and postsynaptic scaffolding protein, glutamate ionotropic receptors, and so on. So there's substantial overlap between these genes implicated by copy number analysis and the GWAS analysis of common SNPs. And in another study, so this is another study looking at lipids, so using an exome chip. So this chip is designed specifically to look at variation on the coding sequences, the exons. And the reason for using this at that time was mostly economical, because this chip at that time was a lot cheaper than the SNP chips that cover the entire genome. The exon only constitute about 3% of the genome. So with this study, we found some associations that are specific to East Asian population. So we detected 14 significant associations that are absent in other populations. And 12 of these actually is because these variants are very rare in Europeans. So that's why they were not discovered in Europeans. And only in 2 of them, the effect size was different. That in 2 of these, the effect size was smaller in Europeans, and one, the effect actually was in the opposite direction. And I think one of the implications is that even though there are a lot of overlap between the genetics of common diseases in different populations, there are nevertheless some differences. And in order to truly understand and make use of the genetics of our own population, we really cannot entirely rely on data from other populations. We need sort of studies done on our own population. Another insight from genome-wide association studies is that there are variants which have more than one effect. There are variants that have multiple effects. And a very good example of this is this gene, TM6SF2, and this gene affects the serum level of VLDL, this lipid. And it does this because it affects the efflux of VLDL from the liver to the blood. So increased activity of this gene, this enzyme, raises blood levels of this lipid, and this increases coronary heart disease risk. But on the other hand, decreased activity would reduce the blood level, but increase the accumulation of lipids in the liver, and this causes increase the risk of non-alcoholic fatty liver disease. So we have to be very careful when we characterize a SNP and say this may be useful for a particular common disorder, because at the same time as benefiting one common disorder, it may be doing harm in another way. So in order to truly utilize this knowledge, we do need to understand the effects of a SNP in a more comprehensive way, knowing what it does in a spectrum of disorders. Another insight is that there are some diseases that are affected by the same genes. And many years ago, we did a twin study, which was in London, colleagues in London, which looked at a number of psychiatric disorders, schizophrenia, mania, and schizoaffective disorder, using a twin design, and we found evidence that schizophrenia and mania had genes in common, the genes that influence the risk of those disorders. And now this has been confirmed by GWAS. So, GWAS has shown that there is an excess of SNPs that are GWAS hits for both schizophrenia and bipolar, you can see here. And indeed, for most pairs of psychiatric disorders, there is significant genetic overlap. And this has been done by another group, Ulrich Sullivan, so looking at the genetic overlap between a range of common disorders. So looking at all pairs of these common disorders and estimating the amount of genetic overlap. And their conclusion is that genetic sharing is greatest for diseases that affect the same cell type or same organ or same system. So perhaps not very surprising. Another use of genetic data is to try to resolve causal relationships. So there's a problem in causal inference that may be confounding. So if we were to try to investigate whether there's a causal relationship between X and Y, then the presence of a correlation is not good enough because there could be a confounding variable. So one way of getting around that is to have an instrument, such as here, I, which we can be quite confident is not directly related to Y, and that the relationship must be, if there's any, must be through the putative exposure X. But this actually is very difficult to be confident that this assumption that the instrument is not related to Y except through X. This is a very difficult assumption to be confident about. And what genetic provide potentially is multiple instruments. So if X has been subjected to a GWAS study, then there could be multiple SNPs which are all associated with X. And so that would provide multiple tests to estimate this relationship, this causal relationship B. So if they are all consistent, then we can have much higher confidence that there is a causal relationship. So here are data using this approach from a group from Iceland. So it plots the effect size of the SNP on lipid levels on the x-axis and the effect size of the SNP to coronary heart disease on the y-axis. And you can see here there is a very clear linear relationship between the effect size on the lipid level and the effect size on the CAD. And so all these different SNPs are giving us a consistent estimate of B, so that this causal relationship we can be quite confident of. And this is for rare variants, and this is for common variants, and this is for rare variants. And they are both telling us the same story, supporting a causal relationship. And in my group, Clara Tang also replicated this. Well, one of the implications of a polygenic model is that the effect size of any single SNP is going to be very small, and so of relatively little value in prediction. Prediction will be very weak. And in order to predict the disease risk, we need to combine information from multiple SNPs. So combining multiple SNPs will give us a score, which we call polygenic risk score, and we can stratify individuals according to the value of the polygenic score. So for example, into deciles, the lowest 10% to the highest 10%. And for schizophrenia, with the current GWAS data, the highest and the lowest deciles have a difference of risk of about 8 to 20-fold. So this is already quite a substantial discrimination in risk, and this is sort of comparable to positive family history. And this is another example of polygenic score, of dividing individuals, stratifying them according to the number of risk values, and looking at the lipid levels for these different strata, and there's a clear relationship. And this can be of use for population screening. We can stratify the population into different decile of risk according to polygenic score. So these different colors represent the different deciles, the lowest risk to the higher risk, and we can estimate the risk over the next with age, with increasing age. And for screening, for breast cancer, there is a recommendation that when the risk exceeds a certain level, then the screening should start. And by stratification, by the polygenic score, maybe we can determine the appropriate age for starting screening more precisely. And of course, we can also take family history into account, because our polygenic score is not capturing the entire genetic components yet. And so by combining the SNP information and the family history, we can get more accurate risk prediction. And another possible area that association studies can help is to predict treatment response. And together with collaborator in Chengdu, Professor Tao Li, we have recently shown using exome sequencing that in patients who are not responsive to antipsychotic medications, they have an excess of rare damaging variants in two sets of genes. These two sets of genes are involved in glutamate neurotransmission, reduced NMDA-mediated synaptic current, and AMPA-mediated synaptic current. And this perhaps makes sense because our antipsychotic at the moment act on the dopamine system, and if there's a problem with the glutamate system, maybe that it makes sense that they are not as responsive to current antipsychotic drugs. And lastly, so in working with my former students, So Cham, we published a paper on using GWAS data to try to reposition current drugs to new indications. So the approach is to firstly to use the GWAS results on the disorder to try to impute the changes in gene expression, because we can refer to the GTEx database, which contain both genotype and gene expression data. So with the GWAS findings, we can work out the predicted gene expression changes. And then we can compare those changes to the data on the effects of drugs, existing drugs on gene expression, to try to identify drugs whose effects on gene expression may reverse the predicted changes in disease. And by doing this, we identify a number of possible new indications for existing drugs. An example is aspirin, and aspirin in our analysis was identified as a possible treatment for bipolar disorder. And indeed, it just happened that recently there was a trial of using aspirin in bipolar, and there's some evidence that aspirin may be useful in a combination therapy. So just to summarize, I've just stated some what I think are the future trends in the genetics of common diseases. So one trend is the increasing importance of large population cohorts with comprehensive assessments of the phenotype, multiple clinical outcomes, intermediate phenotype, biomarkers, lifestyles, and so on. And increasingly, large-scale whole genome sequencing will be used. And this is because, no doubt, rare variants will be very important. And there's relatively less known about rare variants just because they have not been studied very well because of the relative expense of whole genome sequencing compared to single-array. And then, of course, it will be very powerful if we can link up genetic data with clinical databases and other data domains. And increasingly, there's more and more functional annotation data, eQTL, epigenome, and so on. And integrating the functional annotation into the genetic data will be very powerful. And of course, there's also very tremendous developments in functional assays, development of CRISPR and so on. And this will allow us to use these to evaluate the consequences of genetic variation. Thank you very much.